Hello, my name is Noelle Langmack, and I will be presenting today on the French GMO study on Monsanto's Roundup Ready corn and its criticisms and ethical implications. This presentation will include background information on GMOs, the facts on the study and the results, criticisms of the study's data and procedures, effects of the study, the stakeholders, and finally, the ethical implications of the case. GMOs are genetically modified organisms, and genetically modified crops are deeply unpopular in Europe, but are common in the United States, where they have been grown and consumed for more than 15 years. According to the USDA Economic Research Service, as of 2011, 76 to 96% of corn crops in the U.S. had some sort of genetic modification, depending on which state they were grown, even though most developed countries do not consider GMOs safe. This study was written by French researcher Gilles-Éric Seralini of the University of Caen and was published in the journal Food and Chemical Toxicology in September of 2012. It was a 2-year study that explored the effects of feeding rats a high-dose, lifetime diet of Monsanto's genetically modified corn or exposed rats to its top-selling weed killer Roundup. This kind of GMO corn accounts for more than half of the U.S. crop, yet the French team says this is the first time it has been tested for toxicity throughout a rat's lifespan. The study involved 200 Sprague Dawley rats divided into groups. The first group was fed varied diets with genetically modified products without Roundup exposure. The second fed Monsanto GMO corn with Roundup weed killer exposure. The third given Monsanto GMO corn without Roundup weed killer exposure. The fourth given Roundup weed killer in their water, similar to the levels found in U.S. tap water. And the last, the control, was fed non-GMO corn and water without Roundup. All groups with GMO corn in their diets were given the GMO corn as 11%, 22%, or 33% of their diet. Now, what is Monsanto's Roundup Ready corn? It is a genetically engineered variety of glyphosate-resistant corn, and glyphosate is the technical name for Monsanto's pesticide of choice, Roundup. The modification makes corn resistant to changes to its essential amino acid structure when sprayed by the pesticide. The benefits of using this technology include the fact that Monsanto's Roundup Ready corn shows increased yields compared to non-GMO corn. It assists farmers with weed control and decreases the total cost per acre farmers have to spend on their crop. The disadvantages to using Roundup Ready corn are the unknown health risks it poses to livestock and humans, as well as unknown environmental risks and the fact that Roundup weed killer is more toxic than DDT. Additionally, Roundup Ready corn is sterile, meaning it doesn't produce seeds for farmers to use to grow crops for the next season, so farmers have to keep buying seeds from Monsanto and become reliant on this process. Researchers found that rodents fed a GMO diet experienced hormone imbalances, more and bigger tumors, and multiple organ damage than rats fed a non-GMO diet. The GMO or pesticide-fed rats also died earlier. The tumors seemed to be late developing large mammary tumors, and affected rats suffered from severe liver and kidney damage. And though the tumors did not metastasize or spread to other body parts, they were so large they blocked organ function in the rats. 50% of males and 70% of females eating Monsanto GMO corn died earlier compared to 30% of males and 20% of females not eating GMO products. Female rats seem to be more negatively affected by GMO corn diets, whether it was sprayed with Roundup or not, and researchers saw a significant increase in tumors after 1 year. The study received a high volume of media backlash following its release. Not all of the information from the study was published, and scientists found many flaws in Seralini's research methods. Over 700 scientists have signed a petition for the full release of data. Some of the issues they had with the study included the small sample size. The control group of only 20 rats was argued as too small and inadequate to make any deductions from. Corn was the minimum 11% of the rats' diet, which is not a normal diet for them, and invariably distorted the data. Seralini has been funded by Greenpeace in the past, a known anti-GMO organization, which most likely added bias to the study. Researchers also selected a breed of rat that is prone to the same mammary tumors supposedly caused by the GMO diet. Lastly, scientists were confused by the data showing that rats that ate the higher percentage of GMO corn did not get as sick as those that were eating the lower percentages. Further criticisms include comments from the European Food Safety Authority, EFSA. A review panel said that the study's authors failed to establish appropriate control groups as part of the study, that Seralini's study had serious defects in design methodology and does not meet acceptable scientific standards. 6 separate national food safety bodies were also asked to review the study, in France, Germany, Denmark, the Netherlands, Italy, and Belgium, and also came to the same conclusions. One researcher noted that for nearly 20 years, billions of animals in the EU and US have been fed Monsanto GMO Roundup Ready soy products, but no problems have been reported by hundreds of thousands of farmers, officials, and vets. The release of the study also had economic and political effects. For one, the EU and US food safety boards have begun a more thorough look into GMO foods as well as the methodology behind GMO studies. Niki Berlakovich, an Austrian politician, issued this statement. One thing is clear. Given this study, the European Commission has to rethink its verification practices and the approval process must get an in-depth review. On the slightly more dramatic side, France's José Bové, Vice Chairman of the European Parliament's Commission for Agriculture, called for an immediate suspension of all EU cultivation and import authorizations of GMO crops. And thousands of protesters went into the streets of Brussels, calling for an overhaul of food policy in Europe. Michael Antoniou of King's College London voiced the need to test all GMO crops in 2-year lifelong studies. But perhaps the largest effect was on California's Proposition 37 campaign. Prop 37 would require all GMO food in the state of California to be labeled as genetically modified and would have huge fiscal impacts on food producers in the U.S. The study was released 1 month before the proposition was brought to vote, and proponents of the proposition used the study to support their argument. Support for the proposition also included the fact that over 50 countries worldwide, including the EU, China, Japan, and Russia, already require GMO food labeling. Though the proposition did not pass, the study increased its popularity and brought the end vote to 51.4% against Prop 37 and 48.6% for Prop 37. The stakeholders, therefore, are the EFSA and FDA, posed with the task of deciding regulations on GMOs and study results, Monsanto, whose products were under fire, other GMO food companies whose products may have also eventually face criticism, consumers of GMO crops, which includes pretty much everyone in the United States, and countries with GMO food policies, such as Hungary, where GMO foods are completely banned. This analysis brings us to examine the ethical implications of the study and its global effects. First off, is it ethical to release only certain data if it is going to skew the perceived results of a study? I would argue no. Seralini's team did not complete a sound experiment, and not releasing all the data only makes his ethical choices worse. All data should have been published, and when it faced criticism, the researchers should have retracted their conclusions on the basis of distorted and missing data. Next, did Seralini's team strategically release their information at a critical time for the Yes on Proposition 37 campaign? And if so, is this ethically wrong? If they did release the data strategically, then yes, it is unethical, especially because the conclusions are not correct based on lack of data and flaws in methodology. Releasing such a powerful statement could have changed the outcome of voting. The proposition did not pass, but if it had, thousands of food companies in the U.S. and the world would have had to make significant changes to their labeling and risk losing customers. What is Monsanto's role in these findings? Even though the conclusions from this study are obsolete, I believe Monsanto still has the duty to explore the safety of GMO foods and inform the public on the risks of eating these foods. What duties do countries have in GMO food controversies and testing? Each country should use valid data to form their own policies on GMO foods, and I believe it was right of many European countries and the U.S. to review this study and its flaws. To conclude, the final question is whether the study should have ever been published at all. I believe it should have, but with full data and stating that no valid conclusion could be made because of errors in testing methods. That said, some of the data could be useful in further studies for comparison or to help define more efficient and correct studies. Thank you for listening to my presentation, and here are the resources that I used.